Welcome to ISL enabled virtual lab of CSIR.
This tree has a wide spectrum of biological and pharmacological activities. The biological and pharmacological activities of neem, which has 8 different things, which is shown in the picture. The entire plant has therapeutic values as it comprises of complex high number of bioactive ingredients such as nimbine, nimbidine, nimbolide, etc. Anti-carcinogenic properties has been demonstrated by the components of the leaves. Procedure for the isolation and the identification of components of the neem leaves. First, collect fresh neem leaves and dry them. Second, grind the dried leaves into powder and store them in an airtight container. Third, add dried leaves powder into methanol for extraction of leaf components into it. Fourth, isolated the extracted components by performing chromatography. And then fifth, analyze the extracted components by performing mass spectroscopy and NMR. Chromatography is a technique used for separating a mixture of chemical substances into its individual components. This technique enables us to analyze the individual components. Dried neem leaves to be powdered, neem leaves powder in methanol, movement of solutions in the column, mass spectroscopy, analytic technique by which chemical substances are identified by the sorting of gaseous ions in electric and magnetic fields according to their mass to change ratios. Electrobeam source, magnet, detector, sample enters here as shown in the picture. Procedure to check for the bioactivity of the isolated components. First, grow certain type of cancer cells in the laboratory. Second, treat these cancer cells with different concentrations of the isolated compounds. Third, after treatment, perform an assay to check for the isolated compounds' ability to inhibit growth of cancer cells. Fourth, to check for whether the DNA is intact or fragmented due to the effect of the isolated compounds, stain the cancer cells with fluorescent dye and observe these cells under fluorescence microscopy. Fifth, to analyze the cell is in which phase of the cell cycle, stain treated cells with a particular dye. Observe the result using flow cytometry.